GPCRs represent ancient proteins, given that they are present not only in eukaryotes, but also in both eubacteria and archaebacteria. Bacteria have duplicated these genes and adapted them to interact with different substances, including light. Bacteria use rhodopsin to detect light, just as do eukaryotic cells. From the genes which were present in ancestral prokaryotic cells, multiple gene duplications have produced a great diversity of GPCRs in eukaryotes, and this superfamily of genes represents the largest gene family in eukaryotes. Unicellular yeast respond to mating pheromones through G-protein coupled receptors, reminiscent of hormone receptor interactions of animals. Although plants possess GPCRs, they lack many of the GPCRs found in animals, in addition to some of the regulatory mechanisms for GPCRs found in animals, suggesting that many of the animal-specific regulatory mechanisms evolved after the plant lineage had separated. Gene duplications in early animals amplified the number of genes in early animal genomes. For example, there are more than 160 GPCR genes in the genome of fruit flies, which is more than 1%. Nematodes have amplified this even more, with more than 1,000 genes representing more than 5% of the genome. Gene duplications early in eukaryotic history produced large families within this superfamily. For example, family A contains rhodopsin-related proteins, family B contains proteins related to glucagon, parathormone, and calcitonin receptors, family C with metabotropic glutamate receptors, including vomeronasal receptors, family D consisting of pheromone receptors in yeast, family F is known only from slime molds, and then there are additional families as well. And so this superfamily began to produce various lineages of genes which would then further divide to make families of their own.